Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my lecture today. So today we're going to discuss the previous year question of class 12 topics. Sorry, so we are done discussing class 11 topics. So today we'll be discussing class 12 topics. So it is the same paper that we're going to discuss class 12 today. So we have been discussing 2020 year paper, right? So from the same paper we will discuss class 12 topics or class 12 related questions. So it is the year 2020. So we have discussed 46 questions in class 11. So today we will discuss 43 questions in class 12. Yes, ready? So shall we move forward? So come on guys, please do like, share and subscribe the session. So please do share with your friends at least previous year questions. It is important that they know how to solve, so that they get an idea how the questions are coming and how they will approach the question. Alright, shall we move forward? Okay, so without any further delay, so let's begin, because we have to solve 43 questions today in 1 hour. So that's quite a huge task. So I have another announcement for you. So we are meeting once again this evening at 6 p.m. and we are talking about your new chapter, that is cell cycle and cell division. So I am starting a new chapter, so please do join me. So this lecture is at 6 p.m. So this lecture is at 6 p.m. So please join me for the same. So it's a very important lecture, because from this chapter alone in the year 2020 you had 4 questions. So we have to pay attention to this chapter. So please do join me for this chapter. Yes, Madhava Kumar, hello, a very happy good evening. I guess you're new to my session. So please do like, share and subscribe. So thank you so much Madhav Kumar for joining my lecture today. Anthony Abhi, hi, a very happy good evening. So thank you Anthony Abhi for joining my lecture. So please do like, share and subscribe. Okay Anthony, thank you. So moving forward, so I have an analysis for the previous year, that is 2020 question paper. So this is unit wise analysis. So let's see how the unit wise distribution is. So I have defined all the units here. So in class 11 you have 5 unit, in class 12 you have 5 unit. Okay, so the first unit in your class 12 is reproduction, the second unit is genetics and evolution, the third unit is biology in human welfare, then you have biotechnology, and last you have ecology. So you have 4 chapters under this. So that is reproduction in organisms, sexual reproduction, human reproduction, reproductive health. So here you have 3 chapters under this unit. So you have 3 chapters here, you only have 2 chapters here, and you have 4 chapters here. So please analyze how the questions are coming. So yes, Anthony Abhi, I'm good, thank you. How are you? Hi Shimpi, yes, a very happy good evening. I'm good Shimpi, how are you? So thank you so much Shimpi for joining my lecture today. So please do like, share and subscribe. So this is how the weightage varies. Okay, so these are the number of questions. If you can see the highlighted one, that is genetics and evolution, you have a maximum of 11 questions. After that it's followed by biotechnology, you have 9 questions, and you have 2 units which bags 8 questions, right? So first let's discuss genetics and evolution. So let's see what type of questions they have discussed. So this is chapter wise analysis. So individually how many questions are coming from each chapter. So if you once again look from biotechnology 1 and 2, chapter wise you have maximum question from biotechnology 1. So they have concentrated more questions from your restriction enzymes. So we have to pay attention there also. So this is how the weightage varies. And in human reproduction you have 3 questions, and reproductive health you have 2 questions. So together it is 5 questions from this unit. Sorry, from these 2 chapters it is 5. So this is the weightage. So these are the number of questions. So based on this we will discuss the questions now. So first we'll be talking about your genetics and evolution today. Yes, so please give me the, okay, I'm sorry, the answers are here. Okay, yes, see if you know the questions. Yes, so experimental verification of chromosomal theory of inheritance was done by Sutton, Boveri, Morgan, Mendel. Yes, so Sutton and Boveri gave the chromosomal theory of inheritance, right? So they gave the chromosomal theory of inheritance, and Thomas Hunt Morgan, he gave the experimental verification, and he also did what is known as linkage. And Mendel spoke about inheritance of genes, right? So the right answer is Morgan. So here my answers will appear directly. So if you have any issues, so then we will discuss in detail, okay? D, to do something different? No. Okay, so if you know the right answer, don't give me the wrong answer, Shimpi. So directly the right answer is Morgan. Okay? Yes, fine. So moving forward to the next question. So select the incorrect match. So maybe I cannot hide. Okay, yes. So select the incorrect match for me. So please don't look into the options. You have to select the correct match. So phenylketonuria, autosomal dominant trait. Sickle cell anemia, autosomal recessive trait, chromosome 11. Thalassemia, X-linked, hemophilia, Y-linked. So in this question, you can easily give me the answer, but you'll also tell me why the others are wrong. So that is more important. So this shows how well you are in your concept, okay? Because yesterday we have discussed all these diseases. Yes. Tell me which is, so the right answer is already given. So tell me why others are wrong. So what is phenylketonuria? Yes. What is phenylketonuria? Come on. Theoretically by Sutton and Boveri. Yes, you're right. So Sandhya, hi. Yes, a very, very happy good evening. So thank you, Sandhya, for joining my lecture today. So please do like, share and subscribe. Yes. So why? So here the right answer. So the correct match is sickle cell anemia. So here you have an autosomal recessive trait. So it is autosomal and it is recessive. So it is because of the point mutation from GUG to GAG. Right? But why others are wrong? So Satya Seelan, yes, a very, very happy good evening. So thank you for joining my session. So kindly like, share and subscribe, please. So phenylketonuria is also autosomal recessive trait. So thalassemia is not X-linked disease. It is also autosomal recessive. So thalassemia is not sex-linked. So hemophilia is not Y-linked, it is X-linked. So X-linked means sex-linked, right? Yes, you're a new visitor to my channel. Yes, I know. I'm grateful. I'm so very grateful for you joining my channel. So please do like, share and subscribe. So here I am discussing previous year question paper. So I am discussing the year 2020. So phenylketonuria is autosomal recessive. Yes, absolutely right, Madhava. Very good. So here the right answer is option 2. So here we go. Question number 3. How many true breeding pea-plant varieties did Mendel select as pairs which were similar except in one character with contrasting traits? Yes, of course, hemophilia is X-linked. It is sex-linked. So I told you, Queen Victoria's family, Queen Victoria was the carrier. So all the male progeny were suffering from that disease. Yes, Shimpi? Hi Dharani, yes, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Yes, how many true breeding pea plant varieties did Mendel select as pairs? Yes? Yes, come on, what is the answer here? So yes, I have the answer, so that's the main problem here. Yes, so if they ask you how many true breeding pea plant varieties, then it is 14. So if they ask you how many pairs, so that is 7. So you have 7 pairs and 14 traits. So 7 talks about the character and 14 talks about the traits. Okay, so here the right answer is B. Yes, okay. So question 4, the first phase of translation is recognition of DNA molecule, amino acylation of tRNA, recognition of an anticodon, binding of mRNA to ribosome. Yes. So in translation, so we have translation in your central dogma of life. right? So this is the last step in your central dogma of life, where the information on your mRNA is translated to become your proteins. Yes? So recognition of DNA molecule, amino acylation of tRNA, recognition of an anticodon, binding of an mRNA to your ribosome. Yes, the answer is given, but do you know the answer? Yes, so that is the biggest trick. Yes, sorry, yes, what will be the answer here? What is the first phase of translation? So, for translation, your ribosomes have to bind to RNA, right? Your process of translation happens, so this is your translation. So, this happens in your cytoplasm. So this happens in your cytoplasm. So the moment the mRNA comes out from the nucleus, so your ribosomes go and bind. Yes, so then here binding of mRNA to ribosomes is the answer. Okay, clear? So here we go to the next question, that is question number 5. So this is a question that I have solved in my lecture. So if the distance between two consecutive base pairs is 0.34 nanometer and the total number of base pairs of a DNA double helix in a typical mammalian cell is 6.6 into 10 to the power minus 9 base pair, then the length of DNA is approximately. So here what will be your answer? Yes, do the calculation and let me know. Yes, quick. So this is a question which you will not take much time. Yes, quick. So what will be your answer? So how will you solve? So if there are two bases like this, right? So the distance between two consecutive bases is given as 0.34 nanometer. So nanometer is nothing but 0.34 into 10 to the power minus 9 meter. Okay, yes. Then the total number of base pairs in your mammalian cell, so total base pair is 6.6 into 10 to the power minus 9 base pair. Then what is the length of DNA? Yes, Madhava, you're right. So for one base pair, it is 0.34 into 10 to the power minus 9 nanometer. Then for 6.6 into 10 to the power minus 9 base pairs, how many you will have? So if you do cross multiplication, so your total length of DNA is 2.2 meters. Yes, absolutely right. Good. So question number 6. So name the enzyme that facilitates the opening of DNA helix during transcription. So they're not asking replication, they're asking for transcription. So in transcription, what happens? So your DNA, the information on your DNA is getting copied onto your mRNA, right? So here, if I have a DNA like this, so it is double helical DNA, but I write it like this. So 5 prime, 3 prime, 3 prime, 5 prime. But information from only one strand is getting copied. So only this strand's information will be copied to your mRNA. Right? So what is the enzyme that facilitates the opening of DNA helix during transcription? So it has to open like this so that this can be transcribed and you get your mRNA, right? So what is the enzyme? DNA helicase, DNA polymerase, DNA RNA polymerase or DNA ligase? Yes, what is your answer? Yes, what is your answer? So Shimpi says C. Okay, so C is RNA polymerase. So RNA polymerase is the one which catalyzes your transcription. Yes, that unwinds your DNA helix in case of your transcription. In case of your replication, it is DNA helix. So Shimpi, you have given me the right answer. Yes, Madhava, you're also right. Okay, so moving forward to question 7. So which of the following statement is correct? So I want the correct statement. So adenine pairs with thymine through 1 H bond. Adenine pairs with thymine through 3 H bonds. Adenine does not pair with thymine. Adenine pairs with thymine through 2 H bonds. Yes. So what is the bond between A and T? So that is the question. Yes, what is the answer? So Shambhavi, hello, hi. So thank you for joining my lecture today. So please do like, share and subscribe. Yes. Yes, absolutely right. So you have 2 bonds between A and T and you have triple bonds between G and C. Yes, perfectly right. Good. So question 8. So here we go. So which one of the following refers to the correct example of organism? So we are done with Mendelian genetics, that is principles of inheritance and variation. So we are now done with molecular basis of inheritance. So now we are talking about evolution. Okay. So which of the following here refers to the correct example of organism which have evolved due to chances in environment brought by anthropogenic action? So anthropogenic action is man-made or human-made action. Okay. So Darwin's finches of Galapagos Island, herbicide-resistant weeds, then drug-resistant eukaryotes or man-created breeds of domesticated animals like dogs. Yes, please read the question and give me the answer. Which one of the following refers to the correct example of organism, so which have evolved due to the changes in environment brought about by anthropogenic action? Yes? Quick? Yes, what do you think will be the answer? Come on. Yes, any idea? So B, C, D, herbicide resistant, drug resistant. Okay, so you're asking, yes, I'm sorry, yes. So which is created due to anthropogenic action? So you say B, C, D. So according to Shambhavi, the answer is 2. So Shimpi says C. So that is only D. So only D. So Madhava says C. So C is only D. So the right answer is B. So Shambhavi was right. So it is B, C, D. So herbicide resistant weeds we created, right? So we did genetic engineering about a weed which can withstand herbicide. So we repeatedly gave herbicide treatment. So herbicide resistant weeds, right? So if you give antibiotic to a bacteria, your bacteria becomes antibiotic resistant at one point, right? So herbicide resistant weeds also we created. Drug resistant eukaryotes. So we are also becoming resistant to drugs. Drugs does not work on our body these days. Then man created breeds of domesticated animals like dogs. Yes, Shambhavi, you're absolutely right. So the right answer is 2. Very good. Okay. So this was naturally present. This is not due to man-made action. Okay, I'm sorry. Fine. So question 9. So flippers of penguins and dolphins are example of convergent evolution, industrial melanism, natural selection, or adaptive radiation. Yes, good evening. Thank you so much for joining my session. So please do like, share and subscribe. Yes. So flippers of penguins and dolphins are examples of convergent, industrial, natural selection, adaptive radiation. Yes, Shambhavi, you're absolutely right. It is convergent evolution. So here the structure is different, but function is the same. Okay, so industrial melanism is about your moth. So natural selection also supports industrial melanism, and adaptive radiation is your Darwin's finches. So the right answer is 1. It is convergent or analogous. Yes, the structures are analogous and it is convergent evolution. Right. So question number 10. Embryological support for evolution was disapproved by Alfred Wallace, Charles Darwin, so Oparin or Carl Ernst von Baer. So I told you scientist name will not come, but wherever the scientist name is mentioned in NCERT, so I have told you it is important, right? So this is how it will come. So embryological support for evolution was disapproved by whom? Alfred Wallace, Charles Darwin, Oparin, Carl Ernst von Baer. Yes, absolutely right. So Carl Ernst von Baer disapproved this theory because not all higher vertebrates have your gill slit, right? So that is your embryological support. Very good. So one more question. So from his experiments, S.L. Miller produced amino acids by mixing the following in a closed flask. So which of the following did Miller mix? So that is the question. Yes, which of the following did Miller mix in his flask so that he produced amino acids, sugars, etc.? Yes. Yes, what are the things that he mixed in a flask? So ammonia, hydrogen, methane, water vapor at 800 degrees. Yes. Okay. So here it is not methane. So here it is CH3. So CH4 is methane. So this is ammonium. But ammonia was present. So please look into the ions and then give me the answer. Yes, 800 degrees Celsius was absolutely right. So option D is the right answer. So it is methane, hydrogen, ammonia and water vapor at 800 degrees Celsius. It is not A, it is option D. Okay, yes. So can we move forward? Yes. Okay, so we are done with the most important unit, that is genetics and evolution. So we have solved 9 questions. Now moving forward with the next topic or the next unit, this is biotechnology 1 and 2. Okay, so let's talk about biotechnology 1 and 2. Yes, so here we go. Question 1. So in gel electrophoresis, so separated DNA fragments can be visualized with the help of ethidium bromide under UV radiation. Net problem. So is my video buffering? Is my video buffering for everyone? So at acetocaramine in UV radiation, then ethidium bromide in infrared radiation, then acetocaramine in bright blue light. So yes. So in gel electrophoresis, separated DNA fragments can be visualized with the help of, yes Shambhavi, thank you. Yes. So we are talking about biotechnology 1, that is principles and process. So this has almost 6 questions. Yes, so it is ethidium bromide. So you see fragments of DNA like this in your gel. So with the help of ethidium bromide. So ethidium bromide is an intercalating agent. We write it in short as ETBR. So this is viewed under UV light. Yes, absolutely right. So Shimpi, this chapter I have not taught, so you might not know, but very soon we will discuss this chapter. Okay Shimpi? Yes. So the answer is number 1. So question number 2. So the sequence that controls the copy number of linked DNA in a vector is termed ORI site, palindromic sequence, recognition site, selectable marker. It's orange color. Yes, absolutely right Shambhavi. Yes. So the sequence that controls the copy number of the linked DNA in the vector is termed ORI site, palindromic sequence, recognition site, selectable marker. So a vector is something that replicates inside your bacterial cell. So what controls the copy number? Copy number is how many it can replicate into. Yes. What regulates or controls the copy number of the linked DNA in the vector? So Shambhavi says 1, ORI site. Yes, Madhavan, Madhava Kumar, what is your answer? Yes. Others, please type in your answers quickly. So you should not take much time. You should be pretty quick. So, okay. So Shimpi says recognition site. Okay, it's not recognition site, it is ORI sequence. So it how strong an ORI sequence, so that determines the copy number. So copy number is how many copies it is able to produce itself. So to clone a DNA, we do it, right? So yes, it is ORI site, absolutely right. So question number 3. So the specific palindromic sequence which is recognized by eCareerPoint-Eng is. What is the palindromic sequence recognized by eCareerPoint-Eng? Yes. Specific palindromic sequence which is recognized by eCareerPoint-Eng. So eCareerPoint-Eng is your restriction enzyme. So it causes sticky ends, not blend ends. Yes, what is the answer here? Shambhavi says D. Okay, so it is like this. So GAATTC. So it is palindromic, so the same. So CTTAAG. Yes, absolutely right. Very good. Okay, fine. So you are a bit thorough. Okay. So question number 4, identify the wrong statement with regard to your restriction enzymes. So I want the wrong statement here. They cut the strand of DNA at palindromic sequence. They are useful in genetic engineering. Sticky ends can be joined by using DNA ligases. Each restriction enzyme functions by inspection of a DNA sequence. Yes, what is the answer here? So identify the wrong statement. So I want the wrong statement, guys. They cut, so about restriction enzyme. They cut the strand of DNA at palindromic sites. They are useful in genetic engineering. So sticky ends can be joined by using DNA ligase. Each restriction enzyme functions by inspection of a DNA sequence. So everything seems similar, right? So Shimpi says B. Of course, they are useful in genetic engineering, Shimpi. Without restriction enzyme, genetic engineering would not have flourished as it is today. Yes, Shambhavi, Madhav Kumar, what is the answer? So Shambhavi says C. Sticky ends can be joined by using DNA ligase. Okay, so the wrong statement is they cut the strand of DNA at palindromic sites. So why is this wrong? Yes, Madhavan is absolutely right. So this is right. So the sticky ends are joined together by ligases. So this is right. But why is the first one wrong? So restriction enzyme is a broad category. So under this you have two sequences or two classification. One is endonucleases, the other one is exonucleases. So exonucleases does not cut DNA at palindromic site. So these are pretty closely related and only this is wrong. Thus option A is the right answer. Okay, Shambhavi, it is not C, it is A. Yes. So question 5. Yes, do this match the following for me. So match the organism with its use in biotechnology. So Bacillus thuringiensis, Thermus aquaticus, Agrobacterium tumefaciens, then you have Salmonella typhimurium. Yes? Yes? Yes, you are my student. Okay, so I don't understand the last word. Yes, please do the match. Come on, quick. So Bacillus thuringiensis, so that gives you cry protein. So this has to be officially, everything has to be underlined or it has to be italicized. So Bacillus thuringiensis is cry protein. Thermus aquaticus is DNA polymerase. Agrobacterium tumefaciens is your cloning vector. And then Salmonella typhimurium is the construction of the first DNA molecule, if I am right. So cry proteins. So the right answer should be 2 and 3. So Thermus aquaticus is your DNA polymerase. So it should be option 4. No, Shambhavi says option 1. Okay. Yes, okay, I'm sorry. So Bacillus thuringiensis, cry proteins, Thermus aquaticus, DNA polymerase, Agrobacterium tumefaciens is C, Salmonella typh. Yes. So 1 should be the right answer. Yes. So let's look into the answer. Yes, 1 is the right answer. So clear? Yes, Shambhavi, you're right. So 1 is the right answer. So you have matched it correctly. So moving forward to the next question. So question 6. So once again, it is match the following that they have concentrated. So choose the correct pair from the following. So polymerases break the DNA into fragments. So nucleases separate the 2 strands of DNA. Exonucleases make cuts at specific positions within DNA. Then 4 ligases join the 2 DNA molecules. Yes, what is the right answer? So choose the correct pair from the following. Yes, quick. So please find the correct pair. Polymerase break DNA into fragments. So nucleases separate 2 strands of DNA. So exonucleases make cuts at specific position within DNA. So ligases join the 2 DNA molecules. Okay, so the right one is this. Ligases join the 2 DNA molecules. It's absolutely right. So polymerases does not break DNA. So polymerases catalyze the formation of DNA. So nucleases breaks down DNA. So exonuclease cuts at the ends of DNA. So that is why these 3 are wrong and only this is the correct pair. Okay? Right. So we are going into biotechnology and its applications now. So question 7. Which of the following statement is not correct here? Which of the following statement is not correct? So the proinsulin has an extra peptide called the C-peptide. Okay, the functional insulin has A and B chains linked together by hydrogen bonds. Genetically engineered insulin is produced in recombinant E. coli or transgenic E. coli. In human, insulin is synthesized as proinsulin. So which one here is the incorrect statement? Come on, quick. So I want the incorrect statement. So incorrect statement is option 2. Yes, absolutely right, because it is not hydrogen bonds, it is disulfide bonds, right? So this is the wrong statement. Okay. So one last question from this chapter. So here we go. So match the column for me. Yes, match the column. Match the following columns and select the correct option. So BT column. Yes, you're absolutely right, Shambhavi. So A and B is joined by disulfide bonds or sulfide bonds. So BT cotton, Bacillus thuringiensis, adenosine deaminase deficiency. So that is your gene therapy. So RNA interference, it is cellular defense. PCR is used for the reduction of HIV. So A is 4. Yes, so option 4 is the right answer. Okay, so Shimpi, this is how you mark it. So Shimpi has not read about biotech, so very soon I will teach it to you. Okay? Yes, so the right answer is option 4. Very good. So you guys are giving me the answer very quickly. Good. So here we move. So I have one more question. Okay? So BT cotton variant that was developed by the introduction of toxin gene of Bacillus thuringiensis is resistant to fungal diseases, plant nematodes, insect predators, insect pests. So why do we go for BT resistant varieties? So we have BT cotton, we have BT brinjal, we have BT tomatoes. So why do we go for Bacillus thuringiensis resistant varieties? Yes, let me know. Fungal diseases, plant nematodes, insect predators, insect pests. Yes, good evening. Thank you so much for joining my session. So please do like, share and subscribe. Yes. So insect pest, yes, that's absolutely the right answer, because there are insects which feed on the leaves of the plants, thus reducing the yield. Yes, so the right answer is insect pest. Absolutely fabulous. Good. So here we move to the next chapter or the next unit, that is ecology. So ecology has 4 chapters here. So organism and population is the first chapter that we will start with. Okay. Yes, Ansari. So thank you so much for joining my lecture. So please do like, share and subscribe. So the first question from your organism and population. Secondary metabolites such as nicotine, strychnine and caffeine are produced by plants for their growth response, defense action, effect on reproduction or nutritive value. Yes, quick. Secondary metabolites in plants like these, nicotine, caffeine, are produced by plants for which purpose? Yes, it's an easy one. So you have 2 questions from each of the 4 chapters in your ecology. Yes, let me know. Yes, defense action is the right one. So plants have to protect themselves from herbivores, right? So if you have seen, cows and goats do not go near a particular plant because they contain toxic compounds which are very, very harmful for these herbivores. So these are secondary metabolites which are produced by the plant for a defense mechanism. Yes, absolutely right. So option 2 is the right answer. So now moving on to question number 2. Which of the following is not an attribute of population? Natality, mortality, species interaction, then sex ratio, which is not an attribute of the population. So here attribute means features. So natality talks about the birth rate. So mortality talks about the death rate. Then you have species interaction and then you have sex ratio. Yes? Okay, so in a population you have birth rate, you have immigration. So these 2 increase the population density, that is N. So your death rate, your emigration decrease the population density, and sex ratio is an attribute of your population. So species interaction is the odd one out. No, sex ratio is an aspect or an attribute of population because in a population only you have male and female. If you talk about an individual, it is either male or female, you do not have both. Yes, a population has specific sex ratio. So here the odd one out is species interaction. Yes. So the solution is 3. So question number 3. Yes, please do the match. So match the trophic levels with a correct species example in the grassland ecosystem. Yes, so please do the matching. 4th trophic level, 2nd trophic level, 1st trophic level, 3rd trophic level, what would be the answer? Yes. So here what is given? So you have grass, so you have rabbit, so you have crow, you have vulture. Yes, please mark it for me. So which is what? So of course 1st trophic level will be your grass. So because they are the producers, so this is C, right? So C for which one? So only one option is matching, so it will be this. So 4th trophic level, that is last one, is your vulture, and then 2nd trophic level is your rabbit, so this is B, and then 3rd is your crow, right? So here right answer is option 4, but let's check. Yes, we are right. So the right option is option 4. Good. So here we go to the next chapter. So this we are okay. So we have already started ecosystem. So in relation to gross primary productivity and net primary productivity of an ecosystem, which one of the following statement is right? So please go through the statement. Gross primary productivity is always more than net primary productivity. Gross primary productivity and net primary productivity are one and the same. There is no relation between gross primary productivity and net primary productivity. So I want the right statement. And the 4th one, gross primary productivity is always less than net primary productivity. So I want the correct statement here. Come on quick. So please give me the correct statement. Yes. So Shimpi, D is right. How come D will be right? Yes. Yes, 1 is right. So let me explain what is your gross and primary productivity. So gross primary productivity is the amount of biomass produced by plants during the process of photosynthesis. The entire energy obtained in photosynthesis is your gross primary productivity. What is your net primary productivity? So this entire amount that is present with the plants minus your respiratory losses of plants. This is the total energy obtained by plants, but plants also need some energy for their respiration process, right? The aerobic respiration. So this minus your respiratory losses is your net primary productivity. So always your gross primary productivity is greater than your net primary productivity. Okay, so here it is option 1, that is absolutely right. So Shimpi, please don't get confused. So this is the right statement. Okay, yes, so solution 1 is right. So moving further, so question number 5. Which of the following regions of the globe exhibits highest diversity? Madagascar, Himalayas, Amazon forest, or Western Ghats of India? So I am talking about the globe. So this is the world. Yes, what is the right answer? Yes, what is the right answer here? Okay, so the right answer is Amazon forest. Yes, all of you have given me the right answer. So perfect. So this is option 3. Okay, good. So yes, this is 3. So moving forward to the next question. So question 6. According to Robert May, the global species diversity is about 20 million, 50 million, 7 million, 1.5 million. If you're a student who has already attempted 2020 NEET exam, then it should be a very good revision for you because you know where you have made mistakes, right? Yes? But I don't know how many of you have analyzed the paper. So this is how you go around. So first you sort out the paper, you analyze how many questions have come from which chapter. So how should I prepare, etc. So hi Disha, how are you? So you're here. Thank you so much. So yesterday also you joined my session. So thank you Disha. I hope you're doing good. So please do like, share and subscribe. Yes. What is the answer? So yes, Shambhavi says 3. So thank you Shambhavi. So you have been consistent with me since the start of the session. So it is 7 million. That is the estimate given by Robert May. Yes, Madhav Kumar, you're also right. So Shimpi, it is not 1.5 million. So this question I have taught you. So it is 7 million. This is your first attempt. Okay, so good. You're very good. So you're able to give me the right answer. Only once you went wrong, I guess, but you're pretty good. So be consistent. So take a lot of quizzes. So you will be very strong. Okay, Shambhavi? Yes? Fine. So this is 7 million. Okay. So the next question, question number 7. Montreal Protocol was signed in 1987 for the control of emission of ozone depleting substance, release of greenhouse gas, and the other one is your disposal of e-waste, then transport of genetically modified organisms from one country to the other country. So this is from the chapter Environmental Issues. Yes, Montreal Protocol was signed in 1987 for the control of emission of ozone depleting substance, release of greenhouse gases, disposal of e-waste, transport of genetically modified organism from one country to the other. Yes? You haven't studied this topic. Okay, Disha, that's fine. Yes, others who have studied, what is the answer? Quick. Others who have studied, what will be the answer? Emission of ozone depleting substance. So yes, there is a close doubt between the first one and the second one, that is ozone depleting substance and release of greenhouse gases. So Shambhavi, what is your guess? So Shimpi, we have also done this chapter. So what is your guess? So the right answer is emission of ozone depleting substance. So this is your Montreal Protocol. So what are the ozone depleting substance? That is your CFCs, right? So that causes halt in the process of your ozone formation. So it is option 1. So going on to question number 8. So snow blindness in Antarctic region is due to inflammation of cornea due to high dose of UVB radiation. So high reflection of light from snow, damage to retina caused by infrared rays, freezing of fluids in the eye by low temperature. Yes. Snow blindness in Antarctic region is due to. So snow blindness is caused by a particular type of your UV radiation. Yes. Hi Satyendra, you're here after a very long time. So where did you disappear in between? Yes, I'm good. Thank you. How are you? Yes, a very happy good evening. So you're here when I'm about to end the session. But still, thank you for joining my session. So please do like, share and subscribe. So damage to the retina caused by infrared rays. So infrared rays is not bad. Huh? So inflammation of cornea due to high dose of UVB radiation. So UVB radiation is pretty harmful to the eyes. So when you are trekking and if there is a snowfall, so you try to wear goggles because you prevent your eyes from getting blind, because the snow causes blindness to your eye. Okay? Yes. So the right answer is solution 1. So that is option 1. That is the right solution. Okay, so here we move on into reproduction, but I guess okay, so we will continue. So we'll continue with 3-4 questions. Okay, so the plant parts which consist of 2 generations, one within the other. Pollen grains inside anther, germinated pollen grain with 2 male gametes, then seed inside the fruit, embryo sac inside the ovule. So this is from your sexual reproduction in flowering plants. This unit is reproduction. Okay, so you had fever. Okay, so please take care. And please don't fall sick because your third wave is right now here. So we don't know how it will get. So you had a normal fever, but still take care. Okay. So which plant part consists of 2 generations, one within the other? So pollen grain inside the anther. So germinated pollen grain with 2 male gametes, seed inside the fruit, embryo sac inside the ovule. So Shambhavi says C. So seed inside the fruit. So fruit is the generation now. Seed is the next generation, right? Okay, so sorry, I have answers like this. I'm sorry. Yes, now tell me. So now tell me what is the right answer. Yes, guys, come on. So now tell me what is the right answer. So Madhu Kumar says C. That is A and D. Pollen grains inside the anther. Okay. So Shimpi says B, C and D. Okay. Disha says B. Okay. So the right answer is 3. That is A and D. So pollen grains inside the anther consist of 2 generations, one within the other. Okay. So pollen grains inside the anther. So these pollen grains now undergo pollination. They produce male gametes. Okay. So then embryo sac inside the ovule. So this is also a next generation. So your embryo sac is the next generation, right? So that is going to give rise to a new plant, new zygote. So it is A and D. Pollen grains, because we are mainly focusing on the gametes. Pollen grains and embryo sac. Yes? Yes, so this is how the questions come. So this is your NEET 2020 paper. So the right answer is A and D. So they're basically concentrating on your gametes because your gametes gives rise to the next generation. Yes? Okay, so question number 2. In water hyacinth and water lily, pollination takes place by water currents only, wind and water current, insects and water, insects or wind. Yes? In water hyacinth and water lily, pollination takes place by water currents only. So wind and water, insect and water, insect or wind. So Shimpi says insect or wind. So shall we see? Yes, Shimpi, you're absolutely right. So you remembered this chapter so very well when I thought, is that so? Insects or wind. So though they are water plants, they do not have water pollination. So this is a contrasting example, right? Yes, so though they are water plants, they do not undergo water pollination. They undergo pollination by seeds or insect. Yes, absolutely right. Okay, so here we'll saw one more question and then I will end the session. So the body of the ovule is fused with a funicle at the micropyle end, at the nucellus, chelacal end or hilum? Yes. So the body of the ovule is fused with the funicle at which part? So this is also from your sexual reproduction in flowering plants. Yes, let me know. Yes. So Disha says at the micropylar end. So it is the hilum, right, where the body of the ovule fuses with the funicle. So that is your hilum. Okay, so very good. So you have been giving me very quick answers. So you have made this section very interesting. The remaining questions I will continue in the next session. So we have 2 more units, right? So we still have to talk about complete reproduction, and then we have to talk about biology of human welfare. So after this, I will come up with the next year's session, that is 2019. I will discuss the paper. So thank you so much for joining my session today. So please join me at 6 p.m. So 6 p.m. we are starting a new chapter, that is your cell cycle and cell division. So until then, this is me wishing you more and more. So take care and bye.